Let's get started with our webinar today. Get your access in gear. We have been empowering discovery and advancing therapy for the past 25 years, and our mission is really to go together with you and have the scientific research and medicine tools providing you the solutions for gene therapy as well as biomedical medicine. We're really proud to have a product portfolio that spans from research to clinical applications, and this covering not only the immunology and the cancer field, but also stem cells, cardiovascular, neuroscience, and providing all the tools to translate this into the clinical setting. This could be for graft engineering, immunotherapy, or also regenerative medicine. With these 3 decades of experience, we have a fully dedicated team to help you achieve and expand new clinical therapy applications. We support clinical manufacturing by providing GMP reagents and critical raw materials such as GMP lentiviral vectors. Our headquarters is located in Germany, in Bergisch Gladbach, and also in Tetero, where we produce the bulk of our GMP reagents. In the US, we have a lentiviral facility in Gaithersburg, Lentigen, which is a contract lentivirus manufacturer, and also we have in the Silicon Valley, San Jose. It's a new facility there where we are now setting up and really supporting early through pivotal phase GMP cell therapy development and manufacturing. One of the main drivers of our innovation has been the CliniMax Prodigy platform. It has been used in many clinical trials around the world, and the number of users is growing constantly. The CliniMax Prodigy is an integrated system that includes all the different steps that are needed for manufacturing cell products. As an example, we have the T-cell transduction process. It enables you to do the cell selection, activation, lentiviral transduction, further electroporate if wanted, do the culture and the washing, the media feed, and the expansion within one system, and at the end you will get the harvest and formulate your cells. And this is not only for the T cells, we have also developed other applications that can be used in the Prodigy, including stem cell isolation, DC generation, adherent cell culture, or even hematopoietic stem cell engineering. And even if you don't see your wish in this list, you can also have a customized application made specifically for your needs. If your starting material is frozen, or if you have cord blood, for example, there's always a way to adapt the Prodigy to serve your needs. Here you can see an example of how the Prodigy has been used. This is in our facility in San Jose, in which we're using the Prodigy platform with single-use tubing sets in a closed system, and this allows parallel manufacturing. And this is a great example of cutting-edge facility, and we're continually ramping up the capacity to meet the needs in the field as we get more and more. We know, however, that for commercialization success, you need more than positive clinical data, and even more than the receipt of marketing authorization. As reported by others, there are different factors that play into. For example, the health payer reimbursement, the clinical adoption, the supply logistics, and most importantly, the cost-effective and robust manufacturing strategies. So here I would like to expand a little bit more. Why are the cell therapy cost of goods sold so high? This one question comes up when we put together the pricing for a cell and gene therapy product. Why is it so expensive to produce? This is still a challenge for the field, as the cost of goods is rather high, and in particularly for the autologous products where one lot is one patient. The cost of goods may challenge the commercialization of these products despite the medical success that we see. The main reason is that the manufacturing of these products is highly complex and require a high degree of manual labor to produce. Of course, the materials and the capital represent a high portion, but at the end of the day, typically the cost goes up due to the labor component, as you can see in this graph. If the manufacturing can be automated, then the labor component should be reduced, as we can see here. However, there is still a high proportion of the labor needed even in the automated manufacturing. The reason is really on the testing unit operations. Obviously, we have seen that manufacturing cell products is complex, but even in an automated system, there is a fair amount of user interaction. everything has to be performed in the clean room. You can see in the upper part, the process unit operations requires a certain environment, and of course you need materials, equipment, and operators. However, for the testing unit operations, the supply chain has not been truly perfectionated, so some reagents still need to be in a clean environment, and the interaction needed during the process, it's really a lot, and there's quite a lot of room to improve in there. For every lot that we produce, we need to characterize a wide variety of attributes, and this has to be done using separate methods. For example, we have here an early phase CAR T-cell quality target product profile based on the experience we have. For the final product, at least there is a list of expected critical quality attributes across safety, function, and identity. There is a common compendial assays that are used for pretty much every product, and these are fairly straightforward, but become challenging as we try to shorten the needle-to-needle time. Good to say that sterility, mycoplasm, and endotoxin are all performed with compendial methods and largely can be outsourced successfully or also brought in-house, and these are easy ones. But then we go to the ones that do not have the compendial methods, and they require a fairly high development cycle to ensure that they are fit for purpose and appropriate for use as a safety release test, even for an early phase product. For example, replication competent lentivirus. This can be done with qPCR, but current method for lentiviral vectors is a culture or co-culture method that takes 6 to 8 weeks. And this is suffice to say that development alone is a major undertaking. Another one is the vector copy number, also using PCR. It's a little bit more straightforward, but still not to be ignored because it can also be challenging. For the cell count, we're using automated cell counters now, so there are several options in the market, not so much problem, and even with flow, this can be done. For identity, particularly looking at the cell phenotype, for that we use flow cytometry, and this is really the workhorse modality for cell therapy. Transduction efficiency also uses the flow cytometry. The last part that we see here is the product characterization. And for early phase products, they are not really for the release test, but there is an expectation from agencies that we will perform these in phase 1 studies to ensure that development of a relevant potency assay is going to be possible by the time the registration trials and commercialization requires this validated potency assay. It has to be developed throughout the entire process, and for that we use antigen-stimulated cell cytokine release. We can use multiplex, ELISA with flow, cytometry, cytotoxicity using co-cultures, for example. Or there are also other ways to analyze these CAR T cells. Like I mentioned, flow cytometry is one of them, in which we can also measure all the different subtypes. And there, the analytical suite is far more complex and requires a lot of development parallel to this manufacturing process. So as you could see in the previous slide, the amount of QC is really high. And we also see that the amount of hours that it takes to produce these cells, it's very high. So we produce around 40 lots, for example, here across multiple products, and we did an analysis on the different customers that we have. And we can see that QC comprises in between 35 and 40% of the time used. And this is really driving force for the labor component that we talked about before. This is a very consistent profile where QC is always the highest, or at least a third of the whole process. As we are developing these methods, we want to make sure that these methods are fit for purpose, especially by the time that we get to QC. Otherwise, we will spend more time developing them for this purpose while we are in the actual manufacturing, and this is really a challenge for the field. We should aim to be ready by the time that we get there. The key point to consider is that the fit for purpose changes throughout the development cycle. In the early phase, we're looking at feasibility, design studies, small scale, process parameters, and proof of principle. This is used in assets developing R&D in the lab. It could be in academic settings by grad students, in a lab book, not very detailed. But as we mature, we want to characterize this process performance. In some cases, we want to produce data that is IND enabling. Here, the environment changes a little bit. We have to be sure that we do this in a development lab with a more control performed by the PD scientists with protocols and SOPs. By the time we get to clinical, these assays are used to release products for investigational products, also to characterize the product throughout the study and also stability tests that are performed by QC analysts and QC labs and with qualified SOPs. By the time that we get to commercial, these assays are used for release criteria of the approved drugs and looking at quality control to understand whether or not the manufacturing process is successful or not, and these are performed by technicians in accredited facilities and are validated. These dynamic needs need to evolve really quickly throughout the development. One quick note for cell therapy, and particularly autologous cell products, these methods need to be for appropriate intended purpose. In many cases, patients that have been treated with large amounts of prior treatments or have a high disease burden normally will look different than healthy cells. However, for ethical reasons, all development is done with healthy donor blood, and this leads to a very horrible potential gap. It's something to be aware of. Now it can be managed because some of this deceased blood is available from some vendors, and it is highly recommendable to evaluate them to understand these differences. A lesson we have learned in our projects in this CAR T-cell autologous setting is that the starting cell numbers are varied by 2 orders of magnitudes compared to the actual patients. The concentrations are completely different. In addition, the quality is different. There are sometimes presence of blood cells, or there are different components of the cells due to the prior treatments. It really results in product variability. Here we can see a collection of some samples of patients, and it was remarkable to see the differences. You can see on the left, for example, the CD4 percentage. It can have a huge range from less than 1% to 53%. This is really huge range. We should expect high degree of variability, and our assays should also take this into account. For example, you could start augmenting some of the parameters since it really impacts the development, and it's better to tackle it from the beginning, so always have this in mind. A good place to start for your method development and validation guidance would be to follow the guidelines documents. Most importantly, the ICH Q2RA. This dictates the validation of analytical procedures. The ICH is the International Conference of Harmonization for Technical Requirements of Registration of Pharmaceuticals. They have quite a lot of good guidance for you. And also they are a little bit or fairly complicated, and the interpretation is up to the end user to understand and apply to their product. So this is one of the main challenges. For the cell therapy, these documents are still under revision and are still under drafting. So navigating of these guidelines could be challenging. Probably you will enlist or you will try to get some support from some of the companies in terms of consultants or training to be able to navigate these ones. Key to understand, and you will find in all these documents, will be that your assays will have to give an idea or give a very specific feedback on the specificity, quantification limit, detection limit, linearity, range, robustness, accuracy, and precision. I'm not going into all the details for this, but this is something that definitely needs to or requires a lot of investment on your side. Just to give a quick overview on how this analytical method development looks like. As a first stage, you need to make sure that your procedure in terms of the instrument and the methodology, whatever you choose, it has to be selected based on the intended purpose and the scope of your analytical methods. This will save you some headaches if you don't do this from the beginning. After that, you need to make sure that the robustness of the methods already during the early stages is there. Evaluating these will save you time, and you can understand in the beginning the basic methodology, and you can also learn from your prior experience. Finally, then, you will go into the risk assessment stage to fully understand what effect of changes This I'm talking, for example, if you understand what are the different factors or parameters that can affect your method performance. Let's say that you start using samples that are only healthy. What if you use samples that have been stored for more than 48 hours, or if they have different disease, etc. So you can already start to understand if you change your samples, how would this impact your method? As we look into the clinical path for an IND, it is important to understand where the fit for purpose for the method can really cause interruptions to the clinical path and potentially a delay to enter into the clinic. If we look at a standard early phase product coming from a research site into a CDMO, there is a period of knowledge transfer where the methods and the current science is transferred, and the first step is getting the assays running in the analytical suite development. Here we should already have the feasibility and the design studies, and after that then we go into the scale-up process development. Here we have to make sure that we have all the analytical methods in place in order to transfer them. After that, we go into the process transfer of the engineering, where we have the analytical qualification. This tech transfer is one of the most important steps. Once you get there, you probably are enabling faster and easier transfer to the GMP suite. At the last time or the last part on the GMP side, you have your process qualification, and you also have your in-use and shipment studies stability studies, and the long-term stability studies together with the IND submission. So this is a long path in which if you develop your assay from the beginning to fit for the purpose, you could save a lot of time. So you can see that in the past timeline, there are quite a lot of different activities, and you wonder maybe where are the sources of error in this analytics space? Well, there are plenty of them. It could be the reagents, it could be the analysis and the reporting, so really coming from the human factor, the instruments, or also even the protocols that are not well designed. As I mentioned, all of these errors can be avoided if you plan for fit for purpose. And also, the goal of this webinar is to show you how to tackle this. And in the next session, Jakob is going to guide you how to do this.
Okay, thank you, Marta, for the nice presentation and the handover here. And I will take over to go more into details about your setup in cell manufacturing. So, cell manufacturing is a quite complex task, and therefore it is worth it to break it down and look at every single factor one after the other. A typical way to do it are the 5 M's from Ishikawa, which always works good to get behind the issue, find the root cause, and prevent the problems before they occur. Just to reiterate here that we really need to focus on enabling success for analytical methods by evaluating whether or not the clinical components are available across these 5 components. So, given by nature for the manual steps in the assays, the human factor is the most important. And having the right people in place with the right skill set and training at the appropriate setting level is critical at each step of development to ensure that the method can be developed and transferred and qualified and performed for clinical manufacturing as expected. And this requires not only one group, but many groups working across different aspects of the development of the life cycle. Analytical development is requiring research and development scientists. You need manufacturing scientists and technology analytics, which are a focus group to enable the transfer to GMP environment, and then quality control operations who will be performing the assays. It is oftentimes not apparent how much needs to go into to build these teams. Getting ahead of that, having the right steps in place and have built up these teams really is the most critical component. Ensuring that they have time to be trained is a key component of that. Of course, optimal designed instruments with a lot of automation or simple and guiding instrument software can make things way easier. and really help avoiding mistakes. For example, this 21 CFR Part 11 software for the MaxQuant flow cytometry platform really supports to be compliant to the authorities in GMP manufacturing. But even with a well-featured software, you need to consider reasonable time to make sure that everyone is well trained appropriately. Okay, machine. Obviously, machines are critical components. Flow cytometry, being one of the most important modality in cell therapies, is a critical instrumentation decision. One good choice is to use the MaxQuant, Miltenyi's own flow cytometry platform, for all your flow cytometry needs. It combines a really high degree of automation with consistency in a rather small footprint. It has been developed as a tool for cell therapy analysis and characterization, and really is the primary tool for cell therapy flow cytometry in the clinical space. But of course, there are many assays and many pieces of equipment that need to be considered as well. Miltenyi Biotec is known for cell separation, closed system processing, and gentle cell sorting, but doesn't make everything you need yet. For instance, choosing the right PCR platform in addition to the flow cytometry, or the plate reader, or there's a rather long list of potential equipment that could be part of an analytical suite and should be ensured that the requirements for the instrument and the cost needed are anticipated appropriately. This is true even for standard safety assays. For early phase, oftentimes sterility can be outsourced, but there are many developers that would like to shorten the needle-to-needle time using a rapid release testing, for example, the BAC-T system. And here again, understanding the requirements for installation, training, and operation really need to be put in place well enough in advance to ensure that the facility is capable of managing that equipment appropriately. That goes also for standard equipment like the incubators or endosafe for endotoxin testing or other standard safety assays. Material is also critical. So reagents and consumables really need to be, again, appropriate for the question that being asked. And you should really strive for the phase-appropriate quality. So research reagents are okay in research, but will not be successful in the clinical space. It is important to understand supply chain risks, de-risk wherever possible, and ensure that there are as few as possible boutique reagents. And wherever is possible, understand the lot-to-lot variability and stability of the reagents, as this will impact the output of the method even during the development. Standards is challenging for cell therapy, given that most of the products are live cells, whereby standards are fairly impossible to produce, at least in this point of time. This results, of course, in a lack of off-the-shelf options and oftentimes a long lead time for custom products. Planning for this well enough in advance is important. Reference samples, which are critical for all transfer activities, also need to be appropriately developed, and this oftentimes can take more effort than anticipated. So really develop and characterization before they are needed is required. And the FDA will require multiple batches of the reference samples. So planning for that is going to be helpful in the transfer activities. Miltenyi makes an entire line of flow cytometry reagents, including our reaffinity recombinant antibodies and releasable antibodies, both with the benefits of lot-to-lot consistency and background noise reduction, as well as our quite new CAR detection reagents for CD19, CD22, and BCMA, with two-step stainings ensuring bright signal and low limits of detection, which all are developed in particular for cell therapy characterization. Method. Obviously, the method needs to evolve with the intended purpose. And what is going to be appropriate for research study is not going to be appropriate for the clinical release. So you need to iterate the method, and in particular the documentation of that method, to be suitable for the analysis. And again, you really need to put in the time to evaluate for robustness during the development. This is one of the activities that really pays off in dividends the earlier you do it. Align with that. Dedicate sufficient time and resources for review of these methods and training of the analysts. And then implementing trending to continuously assess whether or not the assay is still fit for the purpose. The needs do change over time. One small but really useful thing can be to know the capability of your instruments and using synergies where these exist. For example, the MaxQuant flow cytometric analyzer can also be used as a volumetric cell counter without the use of any counting beads, due to its precision syringe pump drive fluidics. Using hidden capabilities like this can really reduce workflow complexity. Another important point to avoid lengthy development cycles is to use available methods. And here again, Miltenyi can support this through our express modes, whereby we have automated flow cytometry assays built in with the MaxQuant flow cytometry platform. That can allow to access things like immune cell composition, as well as CAR T cell transduction efficiency. Do this with high automation analysis. The key components of these assays are largely automated, like gating, analysis, and readout, and really can shorten the development time for enabling important readouts like transduction efficiency with minimal development. And lastly, all of this has to be wrapped up in the appropriate environment. So the milieu is important, and this probably needs the most lead time of all. Given that developing and in some cases building up, fitting, qualifying the laboratory can be an extremely resource depending activity. It is really critical to define your user requirements exhaustively in before and to ensure you are building the right milieu. This includes sufficient space for all equipment and analysts, appropriate segregation of activities, particularly for autologous therapy, segregation patient blood material, ensuring that the appropriate level of redundancy is in place, so not relying on a single instrument that need to be used for the release. And that the layout really supports an efficient workflow. In some cases, avoiding cross-contamination is critical to success in the QC laboratory. Lastly, accounting for future needs, making sure that your lab design accounts for what you need not only now, but in future. Summarizing, appropriate time is needed to set everything up. Thinking in advance here really pays off. That means especially having the right people in place with the right skill set and training at the appropriate set level is critical. Ensuring that the requirement for the instruments and costs needed are anticipated appropriately. Material really needs to be appropriate for the question being asked, and standards and references samples are challenging and need to be developed and characterized before they are needed. The method needs to evolve with the intended purpose and the phase you want to reach. And then implementing trending to continuously assess whether or not the assay is still fit for purpose. And lastly, all of this has to be wrapped up in the appropriate environment, which probably needs the most lead time of them all. Looking at all of this way in advance can be really helpful to remove the analytical bottlenecks and ensuring the success.